Welcome back to my plant physiology series. My name is Grant Kramer and I am a professor emeritus at the University of Nevada, Reno. I'm standing in front of a new biotech firm called Singular Genomics in the North Campus of UC San Diego, one of many genomics and biotech companies in the area. Today, we're diving into genomics and systems biology, two revolutionary fields that are reshaping how we study plants at the molecular level. We'll explore how genomic data helps us to understand plant traits and how systems biology integrates this information to reveal complex interactions within plant cells, tissues, and ecosystems. This lecture will lay the scientific foundation for future lectures on how plants respond to environmental signals and hormones. You will need to listen to this lecture in order to fully understand what will come next. Before we get started, I would like to acknowledge the great scientists who have significantly contributed to our understanding of the world around us. Today, we highlight two individuals who've had a profound effect on the study of DNA, genomics, and systems biology. Frederick Sanger was a biochemist and a professor at the University of Cambridge. He has won the Nobel Prize for Chemistry twice, something only 4 other people have done. He developed the first DNA sequencing that is still used today. It is called the Sanger method. This method is the gold standard for sequencing and laid the foundation for the newer, faster, multi-parallel sequencing technologies used today. Leroy Hood is a professor at the University of Washington and is the founder of the Institute of Systems Biology. He is a pioneer in biotechnology. He has won countless awards for his research and leadership in the field. He has developed at least 5 inventions that have revolutionized the field in peptide and DNA sequencing. I think he has had even a greater influence in the concept of systems biology and his advocacy for what he calls P4 medicine: predictive, preventive, personalized, and participatory medicine. We are in a biological revolution. The Industrial Revolution started around 1760 with the development of new machinery and factories. The Information Revolution started in the 1950s with the development of new electronics and computers. And the Biological Revolution really got going with developments in DNA technologies in the 1990s and the sequencing of the human genome. This revolution includes the plant sciences, where the Arabidopsis genome was the first plant sequenced in the year 2000, and the grape genome was the first fruit crop sequenced in 2007. The pace of this revolution is accelerating with rapid innovations in molecular technologies and the rapid increase in computer power and artificial intelligence. Here's one way that we are seeing revolutionary changes. The cost of DNA sequencing has dropped dramatically in the last 25 years. I'm not going to go into detail about the sequencing technologies here, but suffice it to say that this is a very dynamic area of research, and future innovations are going to lead to further reductions in cost. I've had my genome sequenced, and it is likely that this will be a commonplace practice for human medicine in the near future. What is genomics? Genomics is the study of an organism's entire DNA sequence, including its structure, function, and evolution. It is the entire set of genetic information and instructions on how to be that organism. In plants, genomic research helps us to improve crop yields, disease resistance, and stress tolerance. Think of the genome as an encyclopedia of instructions on how to make a plant and how to respond to its environment. The grape genome has 19 chromosomes, which represent 38 volumes of instructions on how to be a grape. For example, let's consider this page of genetic code. I want you to use your imagination here. Imagine that the genetic code is made up of a bunch of letters, in this case, in English. It looks like a lot of gibberish. But if we mask most of the letters, we can see instructions buried in the code. In this hypothetical case, it's a set of instructions in the grapeberry to make red color when there's light. The light is sending a signal to the berry to make color in the skin. You can see the message, when there is more light, make more red color in berry skin. Likewise, when the environmental conditions change, a different signal is sent and the grapeberry changes as a result. When there is less light, make less red color in the berry skin. This is a cartoon, of course. In the DNA, there are much more complex sets of instructions. In this diagram I made, I explain the biological processes that make this happen. The plant receives some sort of environmental signal or a developmental cue. In this case, light or rain. This signal is transmitted to the DNA, or programmer, as in the right-hand side, and sends out a message or email known as messenger RNA, which transmits the message to the ribosomes to make particular proteins, which are the factories of the cell, which with the correct supplies make metabolites or products, in this case, the grape colors. While genomics gives us a parts list of genes, systems biology helps us understand how these genes interact with each other and the biochemical machinery of the plant. Systems biology, as seen on the left-hand side, integrates these data from genomics to transcriptomics, or the whole set of transcripts or genes expressed, to proteomics, the whole set of proteins in action, and metabolomics, the complete set of metabolites produced. It integrates them by putting them in comprehensive databases, which enable the building of network models of how the plant functions in different conditions. This kind of integration is only possible with the large computing powers that we have today, and artificial intelligence is greatly enhancing our abilities to analyze the vast amounts of data produced by these analyses. Systems biology is a holistic approach to getting a larger picture or understanding of what is going on. On the right-hand side, we can see that there are several layers or levels of networks that can be considered, from genes to molecular networks, to cellular networks, to organ networks, to the whole organism, or even to the social or community networks. Systems biology is a relatively new discipline. In fact, it is an interdisciplinary approach that integrates a wide variety of disciplines and scientists. Systems biology started about 25 years ago. It started in part because new technological capabilities made it possible for it to happen. The Institute of Systems Biology started in the year 2000. This was near the very beginning of this new field. This systems biology approach made me very interested in the molecular field. Prior to that, I was a whole plant physiologist, and I was interested in the systems biology approach because it provided a holistic approach rather than looking at an individual single molecule protein and studying that. I was much more interested from the physiological point of view of the holistic aspects. I was lucky enough to get funding for myself and my team to be able to conduct research on the systems biology of grapes in 2003, thanks to the National Science Foundation's Plant Genome Program. We were only the second group to be funded for this kind of research. It was the early days as technology was just developing, but progressing rapidly. Before we go further, let's look at the structure and composition of the genome, and then we'll describe the other technologies involved in systems biology in more detail. Genes are buried in chromosomes, which are stored in the nucleus of each and every living cell of our bodies, and each and every cell of a plant. It's the same in every cell, the same copies of the DNA.
Each cell has the complete instructions on how to make that particular organism. Chromosomes are stored in the nucleus and contain chromatin. Chromatin consists of DNA histones and associated proteins. Euchromatin is less condensed chromatin from which most genes are transcribed. Heterochromatin is more densely packed DNA, often consisting of highly repetitive nucleotide sequences known as simple sequence repeats, or SSRs, and transposons. Here we see landmarks that are used by scientists to identify chromosomes. Each row in this figure is an inbred line of maize, and each column is a set of chromosomes. DNA sequences used as probes that are complementary to certain chromosomal landmarks were labeled with fluorescent dyes and hybridized to the chromosome preparations. The green dots seen here are the centromeres near the middle of the chromosomes. The large green area on chromosome 6 is the nucleolar organizer, and the faint red dots are the telomeres, easily seen in chromosomes 2 through 4. The larger blue areas are specific heterochromatic regions. These markers are used to help identify specific chromosomes for various scientific investigations. Besides genes, transposons are found in chromosomes. Transposons are mobile elements in the genome. Transposons were first identified in maize by Dr. Barbara McClintock in the 1940s. Scientists were skeptical of her early observations, but eventually it was recognized that these mobile elements, or jumping genes, were found in almost all organisms. It was many years later that she was recognized for her groundbreaking work and awarded a Nobel Prize in 1983. These transposons are very unusual and unstable. They can move out of the genome and reinsert themselves in another part of the genome. They are also called retroelements or jumping genes. They can alter gene expression by inserting themselves in the middle of the gene and disrupting this so that it cannot be expressed properly, creating mutations in the genome. Most plant chromosomes have a large amount of transposons. In fact, in maize, they make up about 85% of the entire genome. So how does a plant survive such an onslaught? It turns out they regulate or suppress transposon activity by methylating the transposon DNA sequence. That is, the plant has an enzyme, a methyltransferase, that can add a methyl group to a nucleotide. This can block the activity of the transposon and prevent it from jumping into another part of the genome. Pretty cool. It turns out that the same mechanism is also used to control the expression of the plant's own genes. This is known as epigenetic regulation, or known as above the genome or epigenome. Epi meaning above in Greek. This is a new and very exciting research field. So the epigenome refers to a set of chemical modifications that can occur to the DNA and the DNA-associated proteins, such as histones, as you can see here. In addition to suppressing gene expression directly, chemical modifications to the histones can allow the DNA to be unwrapped and allow the genes to be expressed, or it can prevent the unwrapping of the DNA depending on the kind of modification. So epigenetic regulation is quite complicated, and we are only just beginning to understand it.
The epigenome is very sensitive to the environment. These chemical changes or modifications to the DNA can occur in response to environmental conditions. They change the gene expression of the plant and allow it to acclimate to environmental conditions or disease situations. For example, in drought-stressed plants, these epigenetic changes can make the plant more drought tolerant, not only to the current situation, but to situations in the future droughts. In addition, these epigenetic changes can be inherited by the next generation of cells or plants.
So what is transcriptomics? As you recall, it is one component of the systems biology approach. Transcriptomics refers to the entire set of transcripts of a cell or organism. Remember, transcripts are the messages sent to the ribosomes to make a specific protein, most of which are enzymes, the factories of the cell that catalyze chemical reactions to make products for building the plant structure and metabolism. In this figure, we can see an example from my research on grapes, where RNA is extracted from a grape leaf. A sequencing library is produced so that it can be run through a DNA next-generation sequencer. The data from the machine is collected and analyzed to produce figures. The top one displays a heat map of gene expression, or transcript abundance, in 4 different grape genotypes exposed to control well-watered conditions and water deficit conditions. The magenta squares indicate high expression and the blue squares indicate low expression. What we see is that a large number of genes involved in ABA signaling are increased during water stress. ABA is a stress hormone involved in drought stress response. In the bottom figure, you can see more clearly the actual quantitative numbers, showing one genotype having exceptionally higher expression of some of these genes in response to water deficit. This genotype, RM, or also known as Ramsey, is known to be a more drought-tolerant grapevine than the others, indicating that ABA and the ABA signaling pathway are associated with this drought tolerance. Likewise, proteomics refers to the full set of proteins of a cell or organism. Collecting this full set of proteins from a single sample is beyond our current capabilities of proteomic technology. So it is not as comprehensive as transcriptomic approaches today. However, that could change in the future with technological advancements. The transcripts are only the messages to make proteins. The proteins are the actual machines making the products. So in some ways, proteomics is more informative. On the left-hand side, we can see the proteomics workflow. Proteins have to be extracted and prepared before running through a mass spectrometer. In this example, it is an LCMS, which means that the proteins are run through a liquid chromatograph to help separate the proteins before running it through a mass spectrometer. The mass spectrometer is essentially a very sensitive scale that can break up an individual protein into bits and measure the mass of those bits. Then, with a lot of calculations, one can pull all the bits back together and match it to known proteins in a database to identify that specific protein. In addition, the number of bits can be quantified and determine the abundance of that protein. This is shown in the data analysis pane and the result delivery section. On the right-hand side, you can see our research associate, Rebecca Woolsey, who is running one of our mass spectrometers in our proteomics facility at the University of Nevada, Reno. In the bottom right-hand corner, you can see a metabolic map of proteins involved in photosynthesis and photorespiration. In this experiment, I collected proteins from grapevine leaves that had been exposed to water deficit over 16 days. The red squares indicate that the protein responded to water deficit with an increased abundance.
It is clear that these proteins increased in response to water deficit. What's interesting is that these changes preceded the changes in transcript abundance for these proteins, indicating that the proteins were regulated in a different form than from transcriptional regulation, that they're very sensitive to the environment and acting very rapidly. This is a rare insight that is not usually discovered unless one looks at both proteins and transcripts.
As you might guess, metabolomics refers to the full set of metabolites in a cell, organ, or organism. There are only about 2,000 metabolites found in plants. This is much smaller than the 30,000 plus transcripts and proteins found in plants. Even so, it is very difficult to identify all of these metabolites, and different methods are required to separate out different classes of metabolites. Thus, like proteomics, we can only get a partial view of the metabolic profile of the plant, what we might call a targeted approach based on what we are interested in. In the lower left-hand panel, you see a general workflow for metabolite extraction for a variety of organisms. Once the metabolites of interest are extracted, they can be run through a variety of mass spectrometers to identify and quantify the metabolites. Again, this requires lots of data analyses, shown in the bottom right-hand corner. In the upper section of this figure, you can see a GCMS, or gas chromatograph mass spectrometer, that I use to measure volatile aromas from wines grown from well-watered and drought-stressed grapes. These data were collected from 3 different seasons from our experimental vineyard for both Chardonnay and Pinot Noir grapes. The compounds are on the y-axis, and the quantity of the compounds is on the x-axis as a logarithmic scale. So small differences seen on that scale are actually very large or logarithmic. Some compounds were not identifiable and were given a number. Others were identified and given chemical names. Note that a total of about 90 volatile compounds were detected. The arrows highlight specific compounds in the wines that were increased by water deficit. There was hexyl acetate in Chardonnay. Hexyl acetate is a compound that produces fruity aromas. So there were more fruity aromas in wine made from drought-stressed grapes. Likewise, in Pinot Noir, 2-phenethyl acetate and 2-phenyl ethanol were significantly increased by water deficits. 2-phenethyl acetate produces aromas of rose, honey, and apple. 2-phenyl ethanol produces floral aromas. So again, some fruity aromas were increased in Pinot Noir wine that was produced from drought-stressed grapes. So let's summarize this grape systems biology portion. Transcriptomics, proteomics, and metabolomics analyses of drought-stressed grapes gave new clues to how the grapevines were responding. This provided a more holistic picture of the grapevine response. While transcriptomics, proteomics, and metabolomics represent the core of systems biology, a new field is emerging: epigenomics. Epigenomic technology was just beginning as I was nearing retirement. It is a field that is rapidly changing. On the left-hand side, one can see the workflow being used 10 years ago. They're based on short-read technology. Today, there are 2 very long-read technologies from PacBio and Oxford Nanopore Technologies that are advancing the field with much improved detection analyses of the chemical changes to the DNA. It's important to emphasize that all of these technologies capture very large amounts of data, and this data needs to be stored in very large databases before the data can be properly analyzed. These data can then be integrated into other databases and platforms for further analyses. Here are just a few examples of the many databases that are out there. KEGG, as seen in the upper left-hand corner, is a very useful database for overlaying your data onto biochemical pathways. It started with just genes, but other datasets are being added all the time. In the bottom left-hand corner is a database for protein identification called UniProt. At the top right-hand corner is GenBank, a database for genomics data. In the bottom right-hand corner is a reference metabolome database for plants. And in the middle, there is a database called the Plant Reactome. This database is useful for further analyzing molecules like nucleic acids, proteins, complexes, and small molecules that participate in reactions that form a network of biological interactions that are grouped into pathways. Examples of biological pathways in the plant reactome include classical intermediary metabolism, signaling, transcriptional regulation, and developmental pathways. In the future, these pathways will be extended to cover the cell cycle, apoptosis, and resistance to pathogen infections. Another new technique that has emerged with the sequencing of genomes is genome-wide association studies, or GWAS. This technique requires genomic sequencing and massive computer power. Here we can see one kind of output of this technique. It is called a Manhattan plot. It associates certain SNPs, or single nucleotide polymorphisms, with a particular trait. This research method is used by plant breeders to accelerate the development of new plant traits in existing breeding populations. It is a powerful approach that has enhanced breeding efforts for complex traits like stress tolerance and disease resistance. These SNPs allow for enhanced marker-assisted selection for more faster, more precise plant breeding programs. Overall, GWAS has revolutionized plant breeding by providing a high-resolution view of the genetic landscape, enabling breeders to identify and target specific genes or genetic variants to develop improved crop varieties with enhanced agronomic performance. What is a genotype and what is a phenotype? Genotype contains the entire genetic code to make the phenotype. Phenotype is the physical expression of the genotype, including its physical appearance, activities, and metabolism. Phenotype is the product of the genotype and the environment. The environment affects the phenotype through interactions with the genotype. The phenotype is dependent upon three things: genotype, or the genes that determine plant traits; epigenetic modifications of DNA, such as methylation, acetylation; and the environment. Finally, you just can't grind up an entire plant and analyze it. Plants are complex organisms. First of all, no two plants are alike, and there are approximately 250,000 plant species that we know of. Let's look at a simple plant, Arabidopsis thaliana. You can see an example of one in the wild in the upper right-hand corner. Arabidopsis is a simple flowering dicot plant that was used as a reference genome for all other plants because of its short life cycle and small genomic size. It has just 5 chromosomes, consisting of approximately 135 megabases of DNA, with 30,000 protein coding and non-coding genes. To get an idea further of plant complexity, let's look at a young embryo, which only consists of approximately 100 cells. If we look at a young leaf, it contains 100,000 cells. A mature plant, consisting of multiple organs, tissues, organelles, consists of trillions of cells. In fact, most plant traits are very complex as well. Growth involves physiological processes such as photosynthesis, nutrient assimilation, cell production, and expansion. A drought stress response involves more than 6,000 genes, and fruit development involves multiple tissues with changes in genetic programming over time. On the right-hand side, you can see how a grapeberry changes as the fruit develops. In the gray bar, you can see a list of important compounds in the grapeberry, and when those compounds are produced along the time scale. They are not produced at the same time. An important field of research for plant breeders and GWAS is the study of plant traits. This has now emerged into a new field called plant phenomics, the study of plant traits making up a plant's phenotype. Genomics combined with phenomics enhance plant breeding for improved crop performance. You can see an example of some of the plant traits of interest to a wheat breeder in the central figure, and on the right-hand side you can see the workflow that a plant breeder might use to produce a new crop variety. The building of a genome-phenome atlas is underway and will lead to huge gains in our understanding of plant growth and stress resistance. This is a database that can be utilized by plant breeders. So systems biology is not a static field, but is evolving very rapidly. We can now include epigenomics to our systems biology core. On the left-hand side, you can see one workflow that scientists and doctors are applying to human medicine. A hot new topic using this approach involves longevity and the increase of the human health span. Note, I said health span, not lifespan. I am particularly interested in a longer health span. On the right-hand side, you can see the particular workflow that a plant breeder might use today.
Wow, that was a lot, wasn't it? Well, let's briefly summarize here what we talked about today.
This biological revolution is directly linked to technological revolution, especially to DNA technology. I think we can agree that plants are very complex. Computer technology and artificial intelligence facilitate our understanding of this plant complexity. Systems biology is providing a holistic picture of human health and of plants and their responses to the environment.
Well, that's it. I hope you enjoyed this video. If you like this video, then please like it on my YouTube channel and consider subscribing to my YouTube channel, where you'll find other interesting videos like plant physiology, viticulture, and enology, that's winemaking practices. Hope to see you in my next video from Torrey Pines Golf Course in California, La Jolla, beautiful La Jolla, California. Have a great day.